All right, everyone, let's go ahead and I'm going to attempt to do a week 9 virtual lab. There wasn't one that actually exists, so I ended up, I'm going to try to do this just by talking you guys through it. Okay, so in week 9 lab, one of the things that we're trying to do is genetic transformation. And what genetic transformation is essentially genetic engineering. So what we're trying to do is alter an organism's DNA so that it expresses genes that it normally doesn't express. So that's ultimately what genetic engineering is. And there's a lot of ways that we do this in reality. One of the ways that we do this is with extra chromosomal pieces. This is an example of an extra chromosomal plasmid piece. And what we're going to we did in the lab is actually integrate, convert, or take a plasmid extra chromosomal piece and insert it into this organism, E. coli, right? E. coli is just bacteria. It's a prokaryote. So that's one way that it can be done with bacteria. So we can what we're going to do in the lab, or what we did is we actually created a plasmid that allows the bacteria to glow in the dark, actually. And so we have this gene for glowing in the dark that's on this plasmid, and we're going to insert it into this organism. So now that when this organism is in certain conditions, it's actually going to fluoresce green, which is kind of cool. And we're doing it just because we can, and it's a great example of kind of genetic engineering. We can do this also in organisms that are more complicated than E. coli, such as our cells. We actually have other extra chromosomal pieces that can exist in human or animal cells. They're actually called episomes. It's another sort of extra chromosomal piece that kind of looks like a plasmid, but it's specific for animal cells. And then also you can actually use viruses, right? Viruses are another great way to get foreign DNA into a cell. And there's other ways, but those are like the two big ones. Again, so extra chromosomal pieces that look like a plasmid, or viruses are good. All right, so in the lab, what we're doing today, or did rather, is that we had a bacterium, right? This E. coli, and make sure, again, when you're using the name of a bacteria, you either underline or italicize it if it's its Latin name, right? And this little poor unsuspecting bacterium, as we mentioned before, the prokaryote, which means it has a circular genome, which is pretty big and whatnot. But what bacteria also have their capability of is having these extra chromosomal pieces, like we said before. All right, and so what we did is we manipulated the plasmid so that it would do cool stuff like glow in the dark. Now let me redraw this plasmid just so that we can see some of the characteristics on it that made it really cool. All right, so I'm just going to make it in blue. So all plasmids are pretty small. All right, not ridiculously large pieces of DNA. And they all have a couple things in common, and this is, of course, in the lab report for this week, and what make them good as vectors. All right, so all good vectors, or plasmids in this case, plasmids are an example of a vector. A vector is anything that carries foreign DNA into a cell. All right. And what all good vectors have in common is that they have what we call an origin of replication. All right, an origin of replication is a conserved DNA sequence that is going to be recognized by DNA polymerase, and it allows it to be copied once it gets into a cell. So you can actually make multiple copies of this plasmid once it's in. So we could have a bunch of little plasmid babies floating around in the cytoplasm of that E. coli because of the origin of replication. So they're DNA polymerase binding sites, and allow the plasmid to be copied in the cell. All right, hopefully that makes sense. All right, and the shorthanded way of writing an origin of replication is we often write it ORI. All right, so that's the origin of replication. So all plasmids or extra chromosomal pieces like this will have that in common. Another thing that all these will have in common is a selectable marker. And the selectable marker we often use is resistance to an antibiotic in this case, right? So we have to have some sort of almost like a death switch. And that's what the selectable marker does. The selectable marker is going to enable the bacteria, in this case with the plasmid, to survive in a hostile environment, such as an antibiotic that's present in the solution. All right, so all right, so what we're going to do is normally we're going to plate this. Let me go ahead and make it, let's make it yellow. Eventually what we're going to do is we're going to have a plate of almost like jelly. It's like a jelly-like matrix. It's got all the good stuff that the bacteria need to survive, right? And if I have just the broth, just the medium, so the basic medium is called LB, and this is just very basic stuff, right? If I put this bacteria as it is now without the plasmid onto this plate, it is going to grow up nice and happy, right? It's going to be a very, very happy little bacteria, and I'm going to get tons and tons and tons of all those little bacteria growing on that plate, right? Hopefully that makes sense. Now let's imagine that I add ampicillin to this plate. Ampicillin is an antibiotic, and we know what antibiotics do is they kill bacteria. So what's going to happen, of course, in that scenario is if I plate this bacteria onto this plate, hopefully you can see what's going to happen. This bacteria without a plasmid is going to die in the presence of ampicillin, because that's what ampicillin does, is it kills bacteria, right? And so this is not going to be growing on the plate very well. And I'm going to get all those cute little bacteria that I just created, oopsie, are all going to die. The plate's going to be fine. I'm still going to have, again, my nice little jelly-like matrix sitting there, but absolutely none of those bacteria should be able to survive when ampicillin is present, because again, ampicillin is a pretty potent antibiotic. Now let's imagine we have another scenario, the third scenario, and what we did in the lab is now we insert this plasmid into this bacteria. And this plasmid has its origin of replication like we talked about, so it's going to make nice little copies of itself, right? But it also has a gene on it that's a selectable marker that allows this organisms that contain that gene to be ampicillin resistant. All right, so there's a gene, a selectable marker on this plasmid that allows now this organism, E. coli, to thrive in the presence of ampicillin, right? So this is kind of crazy thing that's happened. And so now the bacterium, let me make him, yeah, I'll make him blue. If it's plated onto this plate, we're going to get a ton of those bacteria now growing. Not a ton maybe, but we'll get a lot of them, right? So we're going to get the bacteria now able to survive in the hostile ampicillin environment. All right, so that's why we use selectable markers, is we want to be able to sometimes weed out the stuff that we don't need, right? We only want bacteria that have the plasmid to survive, and so that's why we use the selectable marker. So it's a way of weeding out bacteria that don't have the plasmid. All right, and then the third thing that all bacteria, all vectors rather, are going to have in common, and I'm just going to put this on another sheet so I have a little bit more room. The third thing that they all have in common as good vectors are what we call a multiple cloning site, a multiple restriction enzyme site. Now we talked about restriction enzymes once upon a time. Remember, restriction enzymes are enzymes that cut DNA at very specific sites. All right, so that's going to be really handy for us. So what we're going to be able to do is, if we have this as our plasmid, and let me just redraw it again, we've got our origin of replication, we've got that little ampicillin resistant gene. All right, and I make it red because that's what I was doing earlier. All right. Now what we have is a bunch of DNA sites, like right around here, that we can cut and open up the DNA. And what we do is that when we cut the DNA at those specific sites, let me make it, let's see, let me make it white. All right, so now imagine I could cut the DNA at those sites. All right, now what I'm going to be able to do, I don't know why it's doing that. Let me try this again. Let's see. It's weird that it does it like that. All right. Oh dear. Oh, I don't know why it's doing that, but let me just go ahead and redraw the whole damn thing because it's annoying me. All right, what's going to happen once we're able to cut the DNA at that specific site is that now we're going to be able, let me go ahead and make the circle part again. So now if I cut that DNA at that specific site, I've opened it up, right? So there's where I cut it. And now I can insert actually foreign genes into this area. So now I can put pretty much any foreign DNA into that site, okay? And we do this because sometimes we actually want to genetically alter a cell so that it has a gene in it and now produces a protein in it that it normally didn't produce before. All right, so for instance, let's imagine in this example, what we did this week is the foreign DNA that we integrated into this plasmid was actually a gene that allows the bacteria to eat a sugar called arabinose. Now, normally bacteria cannot eat this sugar, but when we insert this foreign gene, this foreign arabinose gene into the cell, into the plasmid, and then into the cell, it's going to enable the bacteria to now metabolize the stuff that it normally couldn't metabolize. So, and the way I was telling the students to think about it is I cannot eat grass, right? Dr. Rossler is not capable of eating grass, right? But if we genetically engineered me with, say, a sort of like a plasmid construct and injected it into the cells of my gut and incorporated genes in it now that allowed me to eat grass, have the enzymes like cellulase, that'd be pretty cool, actually, somewhat theoretically possible, right? If you put the genes in here to break down grass into my body, right? And then you fed me a lot of grass, I could survive. I would actually theoretically be able to break down now the grass and survive. That's what we're doing to the bacterium today. Normally, the bacteria cannot eat arabinose, right? But because they have the plasmid incorporated inside of their cells in the plasmid, right? Now, if you feed that bacteria arabinose, it can survive because it's now going to be able to break it down because of that gene. All right, so that's what we were doing in this lab today. So that was the big thing. We were forcing this poor little bacterium to be able to eat something it normally can't. Now, that's not that cool, right? We often want to have like what we call the wow factor, right? I want students to kind of get hooked on this stuff. And what we did is that gene, that foreign arabinose eating gene that we incorporated in there, we did one other thing to it. I should have made this a different color probably. What we did is we added a little segment onto it, onto this gene that causes the protein that's then produced to be fluoresce green. All right, so wherever the protein is that's made from this construct, so imagine, so this gene is ultimately going to make a messenger RNA. So this DNA is going to make a messenger RNA that's ultimately going to make a protein to eat arabinose. Let's say this is what the protein looks like. All right. This is the arabinose eating protein. All right. And now that protein actually has a little tag on it that makes it fluoresce in the presence of ultraviolet light. It's going to glow in the dark. So it's just kind of cool. All right. That little tag here is called GFP, green fluorescent protein. And now whenever this plasmid is turned on and eating arabinose and making this protein that allows it to eat arabinose, we're going to see that it glows in the dark. All right. Now, it's crazy, right? That's pretty cool stuff. And we're doing it because we can, and because it's a really good way of kind of showing you guys how cool this stuff is. All right. Let me show you a little bit more about this GFP protein. All right. All right, I think I can get there. I pulled up some pictures. That was my family. All right, so let's see, where is this? All right, so this is GFP. Hopefully you're able to see that. Let me just check. Yeah, good. GFP is derived from jellyfish. All right, so jellyfish and a lot of animals that live in the ocean actually glow in the dark, right? If you actually hit them with fluorescent light, they'll glow. If you've ever been to an aquarium, they often have the jellyfish exhibit. It looks like you're in a rave or something. And you'll see all these kind of funky colors. So literally animals that live under the water, they see in different, quote unquote, see in different wavelengths of light. We see in the visible light's wavelength, and they can see in the ultraviolet light spectrum. And yeah, so anyway, there's proteins in these organisms that allow them to glow in the dark, and specifically often green. So scientists at Stanford actually identified the gene that was responsible for this fluorescence, and it's really small. And they took that gene and now insert it into other sequences just because we can. And now it allows us to see where those proteins that are constructed with the GFP gene go. This is a great example of this. This is actually a mouse, and you can see its eyes glow in the dark, and normally they don't. And what they did here is scientists, there's a protein that's in the retina. So your retina is the back of the eye. And this protein is called rhodopsin, and rhodopsin is what allows you to ultimately be able to see. And so what scientists did when they were first kind of playing around with this is they took rhodopsin and they made it, they labeled it with GFP. So now wherever rhodopsin is present in the body, you hit it with an ultraviolet light, it's going to glow in the dark. Isn't that cool? So here we can actually see that. Here we can see what those, again, the mice. So these mice right here, you can see their eyes have it. They actually, I think this is for rhodopsin again. And we're seeing again that they glow in the dark. This mouse does not have the construct, so obviously it does not glow in the dark. All right, it's just kind of cool. So it allows us to be able to track proteins and see where they exist. Here's another kind of cool picture of this. Here we can actually see in the human body, this is again rhodopsin because I was using it as an example. Here we can actually see wherever the green stuff is, that's where rhodopsin is. And the blue is the nuclei of those cells. And these are actually cells in the eye as well. So it's kind of cool. Yeah, so anyway, we can see that the rhodopsin is kind of spread out, but mostly in this sort of layer at the end right here. All right, and we can see where it is in the eye. And then I think what they did is they treated it with there was some sort of drug that they were using that broke down rhodopsin. So you can see it's very present here, but it's not very present after treating it with this, it looks like a drug for a period of time. All right, and here's some more cool pictures that we can, again, so we use this a lot. And there's also some other wavelengths. Instead of just using GFP, which is what we're seeing here in the green, there's also RFP. Anyway, so we can do lots of cool stuff. All right, so that's what we were doing in the lab today, is we were actually genetically altering the bacteria to eat arabinose and also be resistant to antibiotics like ampicillin, right? And also to be able to glow in the dark when they're doing the metabolism of arabinose. All right, so that was what we did. We actually worked on a lot of lab skills, and so yeah, so yeah, they worked out pretty well. All right, so let's look at our lab report for this week. So if you have the lab template, you can pull that up. And there's some stuff that we're going to get rid of because normally in this week we do PCR, but because of COVID, we're doing that next week. So we're just going to eliminate all the PCR questions, those first, I think, 4. So you can go ahead and delete that. All right, and but we're still going to answer the remaining questions. I help the class actually with this top part, and I don't mind helping you as well. So if you have this pulled up, let me just explain what's happening here. The purpose was to genetically transform some E. coli. So genetic transformation is the introduction of foreign DNA. And E. coli was the organism we were messing with. And the thing that we were messing with is a plasmid. That's what the P is for. All right, so with the plasmid is that extra chromosomal piece. And it was referred to as P glow because it glows in the dark, right? So the P is for plasmid, the glow is, that's a GFP construct. All right. And now this P glow is going to enable to do two things, right? If you're a bacterium that has P glow, you're going to be resistant to the antibiotic ampicillin, right? Normally ampicillin kills bacteria, but if they have P glow inside of the bacteria, they're good. And to metabolize the foreign sugar arabinose. All right, so when they're eating arabinose, they're going to now be able to metabolize this weird arabinose sugar that they normally can't eat. And when they do so, they'll fluoresce green. Fluoresce green when they metabolize arabinose. All right, I was a little bit more detailed in that just because, again, I wanted to make sure you understood what was going on. All right, hopefully that helps you guys to kind of, ultimately understand what we did. It was a fun lab. I thought it was fun. Now we got rid of all those PCR questions, and for whatever reason, I apologize that this is kind of cattywampus. So I'm just going to correct the letters here so that we're all on the same page. All right, so for the first question, it's like, what is genetic transformation? Okay, so what I will encourage you to do is to go into Credo and look up genetic transformation. All right, and it's hopefully you're getting pretty good at using Credo. If you have any questions about how to access Credo, go into your JWU link and click on the library tab, EFG, there we go. And when you get into the library, scroll down a little bit and you should see a link for databases. I'll take you through this. And in the databases, then you can select for Credo. I think I got it all. Did I do it right? HIJK, yeah. All right, so let me just show you how to take you through that. I hope you guys all know this at this point. It's, God, week 9, but better late than never, right? So again, if we go into, let me log back in. All right. Hopefully I'm still sharing this. No, I'm not. I'm glad I checked. All right, so if you go into JWU link, click on library. All right, scroll down a little bit and you can go into databases. And easiest way is just to search for Credo. And it gives you, of course, access then to this ridiculous Credo references, which are like encyclopedias and dictionaries that are all peer reviewed. And let's go ahead and Google for genetic transformation. And there's a couple really good examples. When I was looking, oh, what's a genetic transformation? Let's see. Did I spell everything right? I feel like genetic transformation. Yeah. So composition or structure, these ones are, I don't like that one, I'll be honest. What's going on here? Genetic transformation. Transformation? I don't know. Transformation. I don't know why it didn't show up what I was looking for. This is weird. All right, so our genetic, let's maybe look up genetic engineering. Okay. And this is a little bit better of an answer. This is kind of what we were talking about. It's the idea that we can alter an organism's DNA in a couple different ways. And so just kind of look that up and then hopefully paraphrase it in your own words. You'll need to cite the sources. All right, and so hopefully you're getting good at citing sources. Here's the citation for it, and you can just copy it. I think we're using MLA format or APA, I can never remember. I think we're using APA actually. So you can go ahead and copy and paste that into your reference pages for your lab reports. All right, so you do get into the habit of looking up this sort of material and seeing which answer, of course, kind of corresponds to what you're looking for. And again, it's much better to paraphrase it, kind of try to put it into your own words rather than quoting it. You can quote it, put it in quotation marks, but again, it shows not a mastery of it. I know for my lab groups, I won't give them anything higher than a B if they quote something, but they can get an A if they paraphrase it in their own words. And again, for both situations, they just need to cite it. All right, hopefully you're getting good at that stuff. All right, let's go back to the lab template. All right, let me make sure I'm sharing this. All right, good. And so let's look at the next question. One of the things that we needed to do in order to get the bacterium in, right? So you guys, again, weren't necessarily in lab if you're here, right? So let me just redraw again what we did. And one of the things that we had to do in order to get the plasmid into these poor little bacterium, right, is we needed to we need to make the bacterial cells competent. And what you can cite me on this, and what making them competent is means that they can uptake the foreign plasmid. So remember that we made this little plasmid, right, and it had all this sort of fun stuff on it, and we ultimately want to get this guy inside of that guy. Okay, now to do that, what you need to do is you make the cells competent, and the way that we do it is we treat them with calcium chloride. So we're going to treat the bacterial cells, the E. coli, with calcium chloride. That's just a salt. And what the salt does is it pokes teeny tiny little holes in the membrane. So what we would end up with, let me go ahead and redraw this because unfortunately my eraser worked too well. We're going to get little holes in the membrane. And that's what making them competent, but they're pretty damn small. All right, and so that's why it doesn't kill the cell, right? And also we kept them on ice. And by keeping them on ice, the pores were actually really small because we know things don't move very well, right? There's a lot of, everything kind of sticks together and doesn't spread out very much when things are cold, right? Think of those little penguins that are on the in Antarctica in cold areas, how they're all huddled together. That's kind of what atoms and molecules do in the cold, right? They don't bounce around a lot. They kind of stick together. And so we treated them with calcium chloride. It made these little holes in the membrane, and we kept them on ice so that we kept the holes really, really small. Okay. What we did next in order to get that plasmid in, and it was kind of fun, I think the students seemingly didn't hate it, right, is we did something called heat shocking them. And so what happens in the heat shock is we took those bacteria that were mixed with the plasmid that had the little holes poked into it, and what we did is we heated them up. We made them really hot. And again, what heat does is it makes those holes a little bit bigger, right? So the holes were going to get slightly bigger by heat shocking them. All right. And so that's cool. So we make them nice and toasty, and the holes got bigger because again, what happens in heat is things start bouncing around and spreading around a little bit more, right? And so we do that. And then what we did is we put them on so we heated them up just for about 50 seconds, made the holes really big. And then what we did after that is we hit them back on ice. So by having this drastic temperature fluctuation, it was kind of fun. What ends up happening, hopefully, if we're lucky, is that there's a pressure change. All right. So by going from ice to hot, back to ice, right? What happened is we caused the size of the cell to go, all the way small to all the way big to all the way small again. So we had this, kind of thing happening. And in doing so, there's a pressure change, like almost like a backdraft. If you've ever opened a door to a room or, close the door really quickly, sometimes you feel almost like a breeze. And again, what's happening is there's a pressure differential. And so when that pressure differential occurred, right, what happens is the plasmid that was outside in the solution that we had is going to get hopefully sucked into the cell itself. All right, so all these little plasmid, the plasmid is hopefully making its way into the cell when we heat shocked it. So that's what we did. And it really wasn't that bad. It was kind of fun. Yeah, and so now what we did is then we coaxed those cells to grow, and then we started plating them on those little plates that we mentioned. We have these little plates, and hopefully you'll see them next week if you miss lab. And in that jelly, that this sort of broth, is all the nutrients that helped the bacteria to grow. All right. All right, hopefully that's making sense. Now, when we plated these, so again, let's just remind ourselves of what's on the plasmid. Remember, we had a gene for ampicillin resistance, and we also had this GFP construct that allowed it to eat arabinose. So we had arabinose that was tethered to this GFP gene. All right. All right, and so we coax the cells to start growing and dividing, and so hopefully we get the nice little E. coli that start making copies and copies and copies of themselves, right? All right, so what we did next was we plated a couple of things on, there were 4 different plates that we used, and I'm just gonna go ahead and pull up a whiteboard. Let's see. All right, and let's go ahead and get a clean screen. So let's think about, again, it might be a little challenging for those of you who weren't in lab. We had 2 different test tubes that we were plating, right? We had a test tube here, and here's a test tube here, and I'm going to try to draw all the stuff that was in them. In this test tube, we had what we call minus PGLO. This one did not contain the plasmid, so that's why it's minus. And this one did contain the plasmid, so it's labeled plus PGLO. And in that broth, all right, so we have this sort of brothy-like solution, and actually I'm going to make it yellow because it was kind of yellowy color. And in that yellowy broth, all of them had the bacteria. So this is the bacteria, right? And I'm just going to draw 1 or 2 just for giggles, right? All right, so here are the bacteria. Hopefully there were millions in there, honestly. And so we did some basic like micro work. So those are the E. coli, and I'm just going to label them E. coli. All right. And the difference of what we hopefully accomplished, let's think about it, is that in this test tube, there was no plasmid. So that's all we had. We just had the E. coli. And this is functioning. The reason why we did this experiment is we needed a negative control, right? So we always want to do a negative control, and we always want to do a positive control. Ah, shit. I can't believe I did that. Let me pause it and redraw everything. All right, so I redrew all this stuff. All right, and so what we're seeing here is kind of what we did. We had this sort of jelly-like agar, it's called. It's like a jello. And this jello is pretty awesome. It has what we call LB. LB stands for Luria Broth. So I'll write that on there. Luria, it's the name of, I think, the person who came up with this recipe. And it's like all the vitamins and amino acids and lipids and all that good stuff that the bacteria need to survive is in this broth. Okay, so it's in this sort of jello. So the bacteria freaking love it. All right, so when you work in micro, you'll see these sorts of plates. Now the plate that we see on the left is very just basic. This is literally just plain old stuff, and literally anything will grow on it. Whereas what we see over here in the middle is that this is that broth with ampicillin. So we know, think about what's going to happen, is that only thing that can grow on ampicillin are bacteria that contain the ampicillin resistant gene. All right, so the only thing that's going to be able to grow here is bacteria that contain the plasmid. All right, so let's look at this. So one of the things that we did, and I'll just kind of shoot it first, is that we took this solution and put it on this plate, and then we put, actually let me erase that. And then we took this solution on the right and put it on this plate. Now, hopefully you can think to yourself what's going to happen. Which one of these plates is going to have growth, bacteria growing on it, and one of these plates is not going to have bacteria growing on it. So ask yourself here, is this going to survive here in this medium, or is this going to survive on this medium? All right. Now, hopefully what you thought to yourself is this. This is the one that should have bacteria growing on it, because these bacteria are going to be able to grow on ampicillin because they contain the plasmid that contains the ampicillin-resistant gene. So they're going to be able to thrive. These do not contain the plasmid that confers ampicillin resistant, so they're going to die. So unless you have the ampicillin resistant gene, you are going to die. Bacteria will die on ampicillin plates. Okay, easy peasy. All right, so why did we do this, right? Why did we bother doing this experiment? Is because we wanted to see, of course, we only want, this plate tells me that my transformation worked, that I actually got some of the plasmid getting itself into bacteria. So this is kind of a bit of a positive control, all right? But at this point, we're going to see that, so this one is a negative control too. So we want to see, of course, make sure that we kill, the ampicillin is working. I want to make sure I'm actually killing off bacteria that don't have the plasmid. All right, so this ensures, right, the reason why we did this one is we wanted to ensure that the antibiotic that we're using, the ampicillin, is good, right? It's actually doing its job. All right, so you guys know that drugs go bad over time, right? You might have some Tylenol in your cabinet, right? And you look at it and you look at the expiration date, right? And before the expiration date, it should work like gangbusters, right? But what happens if you take some Tylenol and it's like 5 years old? Most likely it's not going to work very well, right? So that's what this plate, the second plate, is doing for us, is it's telling us, is my ampicillin good? Because I'll be honest, sometimes science doesn't work, right? And this way we're figuring that out, right? Let's think of some other stuff that we did. One of the other things that we did is we took this bacterium, this vial here, and we plated it on this plate. All right, now this is just luria broth. It's just all the good stuff that bacteria love. And hopefully you can make a hypothesis about whether or not there's growth here. All right, you should see a ton of bacteria, right? If everything worked the way we intended it to, we expect there to be a shit ton of bacteria growing on this plate. And again, this is a control. Why do we do this plate is because it tells us whether or not our bacteria that we started off with were even good, right? I want to know whether or not these bacteria are even healthy. Because again, what happens if none of these work? I then have to start from square one and start to figure out what went wrong. But this plate is a negative control, and it tells us that the bacteria, the E. coli, were happy and they were good, right? If we do not see any growth on this plate, that tells us that everything else is pretty much crap, right? Nothing is going to work. All right, so sometimes when you do experiments, you need to build in controls so that at the end of the experiment, if things don't work, right? So we haven't gotten to this plate yet, right? If this plate and this plate and this plate were all empty and this plate, right, we would want to know what went wrong. And by doing these sorts of controls over here, we're able to determine at what point, things happened. So here, again, this was a control that ensured that the ampicillin was good, and this was a control that ensured that the bacteria were healthy. And again, what this did is, again, it screens out only for bacteria with the plasmid, right? So that's the beauty of the ampicillin. The same thing's going to happen here, where the ampicillin is going to screen out only for E. coli that have the plasmid. So that's also going to be the case in this situation. All right. Now, lastly, let's think about what's going to happen here that's different from here. So this plate just has the bacteria with the plasmid growing on them because they have the ampicillin resistant gene. The same thing is going to be happening over here. We're still going to get those bacteria growing, right? So we're still going to get probably the same number-ish of plate of bacteria growing. All right. But now what's different about this medium right here, this yellow stuff, is that it contains the sugar arabinose. Now, arabinose is crazy for this bacteria. Normally, bacteria cannot metabolize arabinose. But remember, what we inserted into this plasmid was a gene that allows them to eat arabinose. So they're going to have a delightful time. They're going to start growing, right? It's like if I had a gene for eating grass, right? And you fed me a ton of grass, right? That gene would get turned on and I would start growing, right? And then what happens as they metabolize it and we hit them with an ultraviolet light, what we're going to see is that these colonies are going to fluoresce green. And again, the reason why they're fluorescing green as they eat arabinose is, remember, they contain the gene for arabinose metabolism plus the GFP construct. So it's kind of fun. All right, so that's the reason. So we had 4 plates, and again, these are the reasons why we use those 4 different plates. And this is ultimately the goal with that last part, right? This is the overall goal of the whole experiment, was to get to this point. And so this allows us to genetically modify the bacteria and have them eat something that they normally can't eat, and when they do so, they fluoresce. All right, hopefully that's helping you guys. So if we go back again to our whatchamacallit. So hopefully you guys were all seeing that whiteboard, all right? All right, so now if we go back to this, and again, here we talked about all of this. And hopefully, I think I've answered all the questions that were here. And I want you to go into explanations for full credit. All right. And yeah, you should be good. And make sure, of course, you put your references. You can reference me for a lot of this, as well as those Credo references that we talked about. All right, best of luck, and ask your lab professors for help if you get really, really stuck.